This presentation is about the role and regulation of glycolysis and gluconeogenesis and aldolases in the cell and various tissues of the body. It explores the specifics of glycolysis and the Krebs cycle. From Grand Canyon University, College of Natural Sciences, Chemistry 530, Biochemistry by Richard W. Cronchi. Aldolase is an enzyme that helps the body break down sugars to produce energy. It's found in high concentrations in the liver and muscles, and when cells containing aldolase are damaged, it's released into the bloodstream. Aldolase levels in the blood can be measured with a blood test to help diagnose and monitor conditions that affects muscles and organs. This test can be used to detect muscle disease, monitor liver or muscular damage, diagnose myopathies, and determine if muscle pain is caused by muscle or nerve problems. Some conditions that can be diagnosed or monitored with aldolase test include muscular dystrophy, polymyositis, dermatomyositis, and autoimmune hepatitis. The normal range for aldolase levels in adults is generally between 1.0 and 7.5 units per liter, but this can vary depending on age and gender. Children under 18 typically have higher levels of aldolase in their blood, with a normal range of under 14.5 units per liter. In organic chemistry, an aldol is a structure consisting of a hydroxyl group OH, 2 carbons away from an aldehyde. The name combines the suffix -ol, OL, from the alcohol and the prefix depending on the carbonyl group, and an ald, ALD, for an aldehyde. An aldol may also use the term beta-hydroxyaldehyde. The term aldol may refer to 3-hydroxybutanal. Aldols are the products of carbon-carbon bond formation reaction, giving them wide applicability as a precursor for a variety of other organic compounds. Aldol reactions in organic chemistry. Aldols are important for organic chemistry and biochemistry because they are intermediates in many key biological and chemical processes. It is widely used in the synthesis of larger molecules, including pharmaceuticals, natural products, and other complex organic molecules. Aldolase in metabolic pathways. Glycolysis and gluconeogenesis. An aldolase enzyme catalyzes aldol reactions in important metabolic pathways like, for example, in glycolysis, aldolase cleaves fructose 1,6-bisphosphate into 2 3-carbon molecules, a dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. This step is essential for generating energy in the form of ATPE. In gluconeogenesis, aldolase catalyzes reverse reaction, playing a vital role in glucose production from non-carbohydrate precursors. Also, aldols and aldol-like intermediates are involved in the biosynthesis of sugars, amino acids, and nucleotides, which are building blocks for RNA, DNA, and proteins. They are also part of the formation of complex carbohydrates and lipids that are essential for cell structure and function. In summary, an aldol compound and reactions are crucial for both synthetic organic chemistry and metabolic organic chemistry in living organisms, making them key players in the construction and breakdown of essential biomolecules. Glucose metabolism is the process by which glucose is broken down to provide energy primarily in the form of ATP, which is necessary for cellular processes. The major pathways involved in glucose metabolism include glycolysis, which is the breakdown of 6-carbon glucose into 2,3-carbon pyruvate molecules yielding ATP and NADH. The end products of glycolysis: 2 molecules of pyruvate per molecule of glucose, and 2 molecules of ATP net gain, 4 ATPs produced minus the 2 ATPs used in the investment phase, and 2 molecules of NADH. Once pyruvic acid molecules are formed, there are 2 pathways. Anaerobic conditions. Pyruvate is reduced to lactate in animals. Lactic acid is formed, awaiting oxygen to further burn or oxidize them for energy, or only in yeast, ethanol is formed. Lactic acid can actually hurt the muscles. Think of carrying a heavy box for a while. Your arm not only gets tired, but starts to hurt. This lactic acid buildup. As soon as you put down the heavy box, the lactic acid in your muscles is immediately converted to pyruvate, which can further be oxidized under aerobic conditions. Lactobacillus eats carbohydrates and poops out lactic acid. More precisely and generally, lactic acid bacteria are acid and salt tolerant, halotolerant, bacteria that digest simple carbohydrates to produce lactic acid. Lactic acid bacteria can be found on decomposing plants, dry products, on the skins of many vegetables and fruits. All of this microbial fermentation is anaerobic, occurring in the absence of oxygen. In a nutshell, lab fermentation transforms sweet, simple carbohydrates into sour acid. Take some produce, chop it up, add salt or brine, put it in a container, cover it, and wait a week or 2. Over that time, acid levels increase and lower the food's pH, giving pickled foods their characteristic pucker. A lower pH also inhibits undesirable microbes that can cause food spoilage and thus preserving food and improving its shelf life. Let's meet the players involved. Yogurt has that sourish taste. Fermentation. Alcoholic fermentation is a biological process that converts sugars into ethanol and carbon dioxide and is a key part of many popular foods and drinks. Yeasts break down sugars like glucose, fructose, and sucrose to create energy for their growth and reproduction. This process is anaerobic, meaning it occurs without oxygen. The process of alcoholic fermentation can be divided into 2 steps. Glycolysis, where the glucose is broken down into 2 pyruvate molecules, which creates energy for the yeast, and fermentation. The pyruvate molecules are converted into ethanol and carbon dioxide. In addition to ethanol and carbon dioxide, alcohol fermentation also produces other byproducts such as heat and water. Applications. Alcoholic fermentation is used to make alcoholic beverages like wine and beer, as well as bread. The flavor of the final product is largely determined by what happens during fermentation. You can see the chemical equation for alcohol fermentation on this slide. Humans have been using alcoholic fermentation for thousands of years, with the ancient Greeks producing mead by fermenting honey and water. Alcoholic fermentation is a complex biochemical process during which yeast converts sugar to alcohol, carbon dioxide, and other metabolic products that contribute to the chemical composition and sensorial properties of fermented foodstuffs. Slide 8, aerobic conditions. After glycolysis, the next step of pathways for breaking down pyruvic acid into CO2 and H2O are the citric acid cycle, which is also known as the Krebs cycle or tricarboxylic acid cycle. This is followed by the electron transport chain. Oxidative phosphorylation is where pyruvate is first converted into acetyl coenzyme A before entering the citric acid cycle. Pyruvic acid is converted into acetyl coenzyme A or acetyl-CoA in a process called pyruvate oxidation, where a carboxyl group is removed from the pyruvate molecules, which is a 3-carbon molecule, releasing carbon dioxide and while the remaining 2-carbon fragment that is attached to CoA forming acetyl-CoA. And the electrons generated during these processes are used to produce ATP in the electron transport chain. The Krebs cycle produces electron carriers like NADH. The electron transport chain is a separate process that uses these carriers to generate energy by moving the electrons through a series of protein complexes within the mitochondrial membrane. The Krebs cycle feeds to the electron transport chain to drive ATP production. Decarboxylation is the first step, and it involves the removal of CO2 molecule from pyruvate, leaving a 2-carbon fragment. Enzymes involved is pyruvate dehydrogenate complex, and it's the enzyme responsible for catalyzing this reaction. Glycolysis is an intricate 10-step process of breaking down 1 molecule of glucose into 2 molecules of pyruvate. It occurs in the cytoplasm and is independent of the presence of oxygen. This 10-step process is broken down into 2 phases with 5 steps in each phase. Phase 1 is the energy investment phase, and phase 2 is the energy payoff phase. Phase 1, step 1, is the conversion of glucose to glucose 6-phosphate by the phosphorylation catalyzed by the enzyme hexokinase in the liver and pancreas. This reaction uses 1 molecule of ATP and trapping the glucose inside the cell. Phase 1, step 2 is glucose 6-phosphate being converted to fructose 6-phosphate using an isomerase enzyme called phosphoglucose isomerase. This again is just isomerization. Phase 1, step 3 is fructose 6-phosphate being catalyzed into fructose 1,6-biphosphate. In this reaction, another phosphate group is added which uses another ATP molecule. Note that this rate-limiting step is regulated by the amount of ATP, AMP, and citrate in a cell. Phase 1, step 4 is fructose 1,6-biphosphate being catalyzed into acetone phosphate and glyceraldehyde phosphate. This reaction is catalyzed by the important enzyme aldolase. And phase 1, step 5, is converting the reactants of step 4 into glyceraldehyde 3-phosphate using the enzyme triose phosphate isomerase. Note that these 5 steps require 2 ATP packets of energy. Phase 2 is the energy payoff phase of glycolysis. Phase 2, step 1, is where glyceraldehyde is converted to 1,3-bisphosphate glycerate by a specific enzyme. And energy net is 1 ATP. Phase 2, step 2, is catalyzed by the enzyme phosphoglycerate kinase to convert the reactant of step 1 into 3-phosphoglycerate in which one other ATP packet is formed. Phase 2, step 3 is catalyzed, the end products of step 2, into phosphoglycerate by the enzyme phosphoglycerate mutase. Phase 2, step 4 uses the products of step 3 into 2-phosphoglycerate by the enzyme enolase. And finally, phase 2, step 5 forms pyruvate by the enzyme pyruvate kinase and produces 2 ATPs. The net products of glycolysis are 2 pyruvates, 2 NADH, and net 2 APEs. 4 ATP are produced, but 2 are used for the investment phase. This next slide shows Krebs cycle. We bring pyruvate into the mitochondria where O2 is present, and we can transition into the Krebs cycle. We generate 2 CO2s and 2 NADHs. Now we are taking acetyl coenzyme A, CoA, and combine it with 4-carbon atom referred to as oxaloacetate, which combines acetyl-CoA. Now we have formed a 6-carbon molecule called citrate. Let me stop here and give you a mnemonic to remember the steps of the Krebs cycle. The mnemonic is citric is Krebs starting substrate for making oxaloacetate. Slide 12. As energy molecules go through the Krebs cycle, we'll start with citrate, which converts acetyl-CoA into oxaloacetate into citrate via the enzyme citric synthase, and is not a reversible step. Just as soon as citrate is made, it is converted into isocitrate. This is just an isomerization reaction. The body can go back and forth between citrate and isocitrate, and the enzyme which does this is called iconotase. See figure A. Isocitrate goes to alpha-ketoglutaric acid, which is a 5-carbon atom, which means that a CO2 is lost between isocitrate and alpha-ketoglutarate. The removal of a CO2 is called decarboxylation. This enzyme is called isocitrate dehydrogenate. See point B on the figure. So then the 5-step carbon alpha-ketoglutarate is decarboxylated and leaves the CO2 behind, where it goes to succinyl-CoA. This is controlled by the enzyme alpha-ketoglutarate dehydrogenase. See spot C on the slide. Now, succinyl-CoA is converted into succinate, see figure D on the slide, via the enzyme succinyl-CoA synthetase. Now, succinate is converted into fumarate. This next step is where fumarate is converted to malate. See point E on the slide. This is done by the enzyme fumarase. Now the malate is then converted back to oxaloacetate. This now completes the Krebs cycle. The enzymes converting malate to oxaloacetate is called malate dehydrogenate. See spot F on the slide. Remember, our entrance into the Krebs cycle in the mitochondria is pyruvate and just one 3-carbon atom. However, glycolysis did produce 2 pyruvates to go through the Krebs cycle. Therefore, the Krebs cycle will cycle through twice for each molecule of glucose. Slide 13. Glucose is so essential for metabolism. However, many times our body runs so low on glucose, yet it still needs some. Gluconeogenesis is a metabolic process that produces glucose from non-carbohydrate sources, such as lactate, glycerol, fatty acids, and amino acids. This is a universal process present in all plants, animals, fungi, and bacteria. In the vertebrate world, gluconeogenesis occurs mainly in the liver and to a lesser extent in the cortex of the kidney. Gluconeogenesis is a key way the body maintains blood glucose levels, especially when fasting or exercise, when many muscles can use fats and amino acids. The brain is nearly exclusively uses glucose. Gluconeogenesis and its pathway. Gluconeogenesis is the process of creating or synthesizing glucose from non-sugar precursors such as amino acids, lactate, pyruvate, and glycerol. This metabolic process occurs in the liver and kidneys and is responsible for maintaining blood glucose level. Gluconeogenesis is critical for the body to maintain blood glucose levels when glycogen storages are depleted, and that can happen during prolonged fasting or intense exercise. It's very important to note that the brain, eye, and kidneys rely solely on glucose for their source of energy. Aldolase is involved in both glycolysis and gluconeogenesis because it catalyzes a reversible reaction. In glycolysis, fructose 1,6-bisphosphate is split into glyceraldehyde 3-phosphate and hydroxyacetone phosphate. In gluconeogenesis, there is a reverse reaction than in glycolysis. So therefore, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are combined together to synthesize or make fructose 1,6-bisphosphate. Aldolase is essentially acting as a key enzyme at a crossroad between the two pathways where the reaction can proceed in either direction depending on the cellular needs. Key points on aldolase's reverse reaction. Number one, it's reversible. And number two, the primary function of aldolase is to cleave a carbon-carbon bond in fructose 1,6-bisphosphate, which can be reversed to join two 3-carbon molecules back together, making it crucial for both the breakdown, glycolysis, and synthesis, gluconeogenesis, of the all-important sugar molecule, glucose. Aldolase is found in the cytoplasm of the cell. Aldolase is an enzyme that exhibits tissue-specific expression patterns. These patterns play a crucial role in both glycolysis and gluconeogenesis. There are three main forms of aldolase in mammals: aldolase A, aldolase B, and aldolase C. All aldolases A, B, and C have distinct physiological roles. Aldolase A, or Aldo-A, is located in muscle tissues, specifically skeletal muscles and heart, as well in erythrocytes. Aldolase A is used in glycolysis and is tailored for tissues with high energy demand, like muscles. It also has a role in an anaerobic glycolysis in red blood cells. Aldolase B, or Aldo-B, is located in the liver, kidneys, and small intestine. Aldolase B plays a critical role in catalyzing the cleavage of fructose and is essential for both gluconeogenesis and fructose metabolism. And aldolase C, or Aldo-C, is located in the brain and nervous system and retina. Aldolase C supports glycolysis in these tissues and is specialized for brain metabolism and neural functioning. Slide 16. Aldolase B, an important enzyme in glycolysis and gluconeogenesis, is primarily regulated when mRNA is copying the code from the DNA. There are 4 transcription factors. The first transcription factor is hepatocyte nuclear factor 1, or HNF1. HNF1 is a key regulator of liver-specific genes, including aldolase B. It binds to the promoter of the region of aldolase B gene and activates transcription in hepatocytes' liver cells. This ensures that aldolase B expression is liver-specific and supports metabolic processes like gluconeogenesis, which are critical for maintaining glucose level. The second transcription factor is hepatocyte nuclear factor 3, or HNF3. HNF3 has been shown to interact with the promoter region of genes, and the promoter region of genes and DNA is a spot or the place on the DNA as it starts to get copied or transcribed by the mRNA. HNF3 controls the binding of the messenger RNA, and it actually provides a binding site for transcription. Slide 17. 2 other transcription factors are C, EBP, and D-box binding protein. In the liver, aldolase B enzyme is promoted through metabolic signals such as fasting or glucagon release. C, EBP contributes to the activation of gluconeogenesis by increasing aldolase B levels during periods when the body requires more glucose production. These are daytime transcription factors. The last transcription factor is DBP, or D-box binding protein, and it is a transcription factor involved in the regulation of the circadian or daily rhythms. D-box promoters align the timing of gene expression and aldolase B production with these circadian rhythms. D-box binding proteins influence gene expression during fasting or nocturnal periods when gluconeogenesis is more active. In summary, HNF1, HNF3 are mainly regulating the liver-specific expression of aldolase B. With CEBP, this amplifies aldolase B's expression in response to metabolic demands, and D-box binding proteins ensure proper alignment with circadian rhythms. Together, these transcription factors finely tune aldolase B levels to meet the liver's metabolic needs in different physiological states. And finally, slide 18. The regulation of glycolysis and gluconeogenesis is a highly coordinated effort that ensures that these opposing pathways do not operate simultaneously at high rates, avoiding a futile type of cycle. Both pathways are controlled primarily by the level of key regulatory enzymes, and their regulation is influenced by the cell's energy needs and hormonal signals. Thank you very much for listening to this presentation.